all of you know that we generate a lot of waste while we are working in laboratories and hospital, and now we focus on the waste that is mainly on the biological samples. Okay, so what can be considered waste? So bila you all tengok so what can be considered waste is that from this image that you all can see that what can be considered as waste here. Okay, so the chocolate wrappers, this is a table where a meeting is going on. So this chocolate wrappers that is being used is a waste. The plastic bottle is a waste, but the plate is not a waste. Okay, it is for recycling purposes. So same thing that for when you look into biological sample, that we need to identify what is going to be our waste and what we need to discard and what we are not going to discard. Okay, so when you look at this one, is what type of waste is that? We call this as domestic waste. So this means that how you are going to segregate your waste. So benda-benda yang terletak dalam beg hitam ni is domestic waste, and benda-benda yang terletak dalam beg kuning ni now is considered as biohazard waste. So in this lecture, we are going to be focused and looking into how that we can manage our biohazard waste. Okay, so if you all have any questions or anything, you all can interject or put it in the chat. Okay, so when we look into waste management, I think this image have gone through quite a viral. So what does it mean here? Okay, if you know about the scenario, this image was captured during the World Cup. Okay, soccer World Cup, football World Cup that happened in Qatar 2022. So what happened is that somebody is texting tiada suara. Can the host check on this? Can you all hear me, some of you? Okay, dengar saja. Nah. Okay, good. I think siapa yang tak dapat dengar tu, you need to check on your internet connection. Sorry. Okay, so if you look into this image, why the purpose that I put this image is that when you look into, all of these are Japanese fans who came for the football match, and after the match, do you see what they are doing? They are clearing up their rubbish. So benda-benda waste yang mereka generate, mereka clear. Okay, they are clearing up their waste. So what does it mean here? So in the lab, any waste that has been created by you are the person that is responsible to clear it. Bukan kata orang lain akan tolong buat untuk kita. So waste management, and especially in biological waste management, is very important that the person who created the waste is the person who need to manage the waste. So dialah yang kena manage waste tu, sebab only you know what waste that you created, what kind of risk that it is in the biological specimen. So benda tu sangat-sangat penting untuk kita tengok. So with that, okay, sorry image ni dah pergi langgar pula. Okay, so this supposed to be a group discussion, tapi I want you to think about that as many that as you can, and if you can put it in the chat, I give you about 1 minute. Okay, what you can write as an example, okay, as waste that you considered biological ataupun infectious. Okay, sorry Okay, I don't know why it becomes like that. Okay, okay, so just put in the chat so that we have some kind of interactive session. Bukan saya seorang yang cakap pun. Okay, good. Culture plate, blood. Okay, tips, petri dish. Okay, patient samples. Lagi siapa yang boleh? Okay, use capillary tube, human urine, patient waste, syringe needle, animal waste ada, carcasses betul, pus, use reagent, okay, PPE, tissues, microbe, sputum, glove, nasal swab, mask, okay. Tapi great, memang banyak yang you all kata, and the list is quite extensive yang you all cakap. So the best thing would be is that kalau somebody said tissue, okay, tissue tu, kalau tissue tu you gunakan untuk lap tangan lepas kamu dah basuh tangan, is it a infectious waste or biological waste? No. Yes. So jangan pergi buang tisu tu dalam yellow bin. Okay. So but the tissue that you use to wipe the bench, okay, you spray dengan alcohol, you wipe the bench lepas kamu kerja, tissue tu is considered infectious and biological waste ke bukan? Yes. Tissue yang kamu gunakan tu is considered as biological waste. Okay. COVID test kit is infectious waste sebab kamu pakai sputum walaupun dia negatif. Okay, you are using your saliva, so it's bodily fluid, biological sample, so dia memang considered infectious. Dia tak keluar result COVID positif, tapi it can keluar influenza positif. Okay, so we look into the high risk. Kita selalu letakkan when we talk about biological infectious samples, we put it as a high risk. Okay, so when you want to categorize biological waste macam kamu listkan tadi, so you ada solid waste which is the non-sharp, then kamu ambil sharps, then you have pathological waste, liquid waste and mixed waste. So solid waste kamu adalah kamu punya macam kamu orang list down tadi daripada tissue, mask, okay, agar, petri dish and everything would be in solid waste. Sharps like syringes, okay, needles dengan syringe, scalpel, semua tu akan jatuh dalam sharp. Okay, so sorry, I think something happened. Okay, so here akan dikira sebagai sharp, and pathological waste adalah macam amputated body parts, ataupun bila kamu buat animal study, kamu ambil lung, liver ke semua apa, so pathological waste. So liquid waste ialah selalunya bacteria culture, cell culture punya media, bacteria culture punya media, all of this which is in the liquid form is liquid waste. Mixed waste adalah campuran, okay, campuran di antara contohnya chemical dengan biological, okay, ataupun radiological dengan biological. So then how you, after that, lepas kamu classify, then only you would know how that you can treat it, how you can disinfect it sebelum kamu nak buang. So categorizing the waste is very, very important. Okay, so good that, kejap, need to open my chat. Okay, so ada orang tanya soalan, what about plastic test tube use tips? Some said must in sharp bin, some said in yellow bin. Okay, plastic waste memang dalam yellow bin, dia bukan sharp. Use tips tu bukan sharp. Okay, dia hanya tip plastik saja. Test tube semua tu bukan sharp. So it has to be in the yellow bin. Okay, it has to be in the yellow bin. Okay, so who defines? Okay, sekarang siapa yang define? Okay, biological waste dalam di Malaysia ni siapa yang define? Siapa yang decide apa yang kita kena buat pada biological waste? Okay, KKM ke? Sebenarnya tak. It falls under the Environmental Quality Act. So, akta yang tengoknya Environmental Quality Act, betul, DOE. Okay, very good. So, DOE yang regulate dan dia jatuh kepada prescribed environmental quality schedule waste treatment and disposal facility. Okay, dia punya amendment pada 2005. And dia pun dah ada guideline on handling the management of clinical waste. Dah ada dah guideline dia. How to manage clinical waste. Okay, dah lama dah, 2009 dah keluarkan guideline. So this guideline is available online. Siapa-siapa yang tak tahu, just you can go and download this guideline and go through the guideline. Okay, apa yang kita dah ada. So sebenarnya orang yang kerja kat hospital tu, they are more aware of this than orang yang tak kerja kat hospital. So kalau kita tengok definition of biohazard waste, okay, so ini adalah definition daripada WHO. Okay, biohazard waste also called infectious waste such as blood, bodily fluids and human cell line. Okay, pun akan terlibat juga dalam ni. Is waste contaminated with potentially infectious agent or other material that are deemed a threat to public health to the environment. So you tengok, deemed threat to the public health or the environment. Okay, jangan tengok public health untuk human saja. Untuk environment dia maksudnya melibatkan animal, plant dan juga environment. Okay, faham ke? Hopefully you understand that. So biohazard waste is waste that has been in contact with any kind of infectious agent. Okay. So what is an infectious waste? Kalau kita tengok ialah isolation waste. Waste generated by hospitalized patient who are isolated to protect others from communicable diseases. And itu memang, okay, you all know because he has been isolated. Culture and stock of infectious agent and associated biological. Contohnya specimen from medical and biological laboratory. Culture and stock of infectious agent from clinical, research and industrial laboratories. Okay. And dia juga ada human and blood products. This dia akan include pada waste from blood, serum, plasma and blood products. So walaupun it is a common routine blood checkup, okay, medical checkup yang kita draw blood and you assume the patient is healthy dan lepas test pun tak ada apa-apa problem, but you cannot buang benda tu kata oh it's not infectious. Okay, dan benda tu dah jadi biological specimen yang you suspect dan you nak test, dia kena dibuang sebagai infectious waste. This is a grey line yang orang selalu argue. Okay, especially people who are working in the laboratories. Kenapa I'm saying, because hospital they have strict guideline and SOP what they need to do. So memang mereka benda ni semua akan dibuang. Tapi orang yang kerja dekat makmal yang you ambil blood, kata you ambil healthy individual sample and then tak ada infectious, you nak buang, tak boleh. Okay, it has to be, even though you have processed it, it has to be dumped it as infectious waste. Okay, so pathological waste macam saya kata tadi is tissue, organ, body part, blood and body fluids. Contaminated sharps, okay, hypodermic needle, syringes, scalpel blade, pasture pipette and broken glass. Okey, di sini saya nak tanya soalan. Kalau kamu ada broken glass, okay, and pasture pipette yang gelas, boleh tak kamu buang dalam sharp bin? Okay, boleh tak kamu buang dalam sharp bin? Sorry. Somebody said no. Somebody said yes. Okay, boleh. Ramainya boleh. Sebenarnya tak boleh. Okay, broken glass tak boleh dibuang dalam sharp bin. Okay. Kenapa? Okay, later I will tell you kenapa. Okay, tak apalah. Okay, kenapa dia tak boleh buang dalam sharp bin? Because all of this will be sent for incineration. Okay, kalau kamu dah buang broken glass dalam tu, and then they send it for incineration, gelas tu akan melt dan akan rosakkan incinerator. Okay, so that is why broken glass cannot be dumped into sharp bin. So apa yang kamu kena buat kalau ada broken glass, you have to collect all the broken glass dan kena treat dalam disinfection solution. And then you collect the broken glass, dispose it as broken glass. Dia ada orang yang akan ambil broken glass tu, and then dia akan recycle it and they will make it back to glass. Okay. How to dispose syringe without needle? Kalau syringe tu tanpa needle, dia bukan sharp. Dia boleh dibuang dalam yellow bin. Kalau tanpa needle, you are using syringes for different purposes, tanpa needle, it goes to the yellow bin, bukan sharp bin. Hanya syringes dengan needle sahaja dia masuk dalam sharp bin. Okay, so kena faham ni. Okay, so don't go and throw things dalam benda yang bukan-bukan, especially broken glass. Sebab tu saya letak kat sini broken glass ni, and how you need to treat the broken glass is totally different. Okay, hope you understand that. And animal carcasses, animal bedding kena buang juga. Okay, dalam hospital no broken glass, only biological waste, sharp chemical waste and general waste. So broken glass, you have to create this now sebab you dah faham. So you have to create this now, and then broken glass tu kamu boleh collect dan simpan, dan dia ada orang yang dah boleh datang ambil. If you need information, I can give it to you. Okay, so you can contact the person, dia akan datang ambil free je daripada kamu. So broken glass need to be collected as broken glass. Kalau you letak dalam sharp bin, benda tu pergi ke incinerator, kesian dekat service provider tu, incinerator dia lama-lama akan accumulate banyak gelas silika ke bawah, dia akan rosak. Okay. So miscellaneous contaminated waste, this includes waste from surgery, autopsy, laboratory waste, dialysis unit waste and contaminated equipment juga. Kamu kena, kalau equipment tu kamu nak guna balik tapi dia dah contaminate, you have to disinfect. Okay. Ataupun memang equipment tu kamu nak dispose, kamu kena disinfect dulu sebelum kamu nak dispose. So benda tu penting. So sebab tu if you realize, I'm keep on repeating, before you need to dispose, you need to disinfect dulu. So benda tu sangat-sangat penting. Even sebelum kamu nak hantar, okay, yellow plastic tu kamu kena autoclave dulu sebelum kamu kena bagi dekat service provider. Except for hospital, where that the service provider comes and collect it every day usually, so mereka tak akan disinfect sebab banyak sangat. So they cannot manage disinfect. So they have a different flow of work yang semuanya dihantar kepada the place where they collect it outside the building, dan benda tu akan declare every day. Okay, tetapi dalam makmal, you buang dalam dalam yellow bin tu sampai kadang-kadang seminggu ke. So benda tu you kena disinfect dulu sebelum you nak letak dalam yellow bin dan you tunggu masa the service provider comes and collect it from you. So that is very important sebab bila kamu tak disinfect, kamu letak kat luar, lama-lama benda tu akan start to grow bacteria dan benda tu akan jadi hazardous kepada service provider yang datang collect tu. Okay. So if you look, who is at increased risk if waste material contaminated with biological material are not handled and treated properly? So untuk soalan pertama ni, siapa yang who's at risk? Siapa yang berisiko kat situ? Siapa yang berisiko kat situ? Any answer? Cleaning staff, user, cleaner, staff dan pekerja kebersihan. Okay, waste handlers. Okay. Kalau waste tu tidak dihandle secara betul, okay, tidak dihandle secara betul, the first thing, okay, the first thing yang akan jadi adalah kamu sendiri. Okay, if you, the worker itself, orang yang bekerja itself akan ada isu ni. Okay, dia sendiri yang akan kena infection first and then orang-orang dalam makmal tu akan kena infection yang bekerja. Ini yang disebut sebagai laboratory acquired infection, LAI. And then as you said, cleaners, the person who's handling the material, semua-semua tu akan ada effect. Okay, saya nampak soalan banyak on glass slide and things. I will come to that at the end. Okay, saya akan jawab soalan tu nanti. So what are the potential incident that can occur? Okay, so apakah potential incident that can occur is LAI lah. Kalau benda tu tak di-treat secara betul dan tak di-maintain, so benda yang akan berlaku adalah LAI. Okay, eruption and spread of infection, betul. Okay, so this one is quite common, benda yang boleh berlaku kalau benda tu. So it is not just handling the sample itu penting, but handling the waste pun penting. Sebab you katakan dalam makmal you ada yellow bin. Okay, lepas-lepas culture you tak disinfect, you buang macam tu saja. Okay, so benda tu dalam tu akan start to grow. So, bila next person nak pergi buang sampah ke apa, dia buka tu, and then if it is a sporulating punya agent, dia akan sporulate dan dia akan fly. So orang tu akan kena infect. Okay, you pun kerja dalam makmal tu juga. So everything has to be treated properly before kita nak even letak dalam waste bin tu. So that thing is quite important yang you all kena faham. Okay, biological waste management bukan kata oh benda tu waste. Tapi sebelum dia nak jadikan dia waste tu, you kena treat dia dulu. Okay, so that is very important. So kalau kita tengok why we need to manage hazardous waste. Okay, it's very simple. Sebab kita nak protect. Okay, sebab kita nak protect human dengan environment. Okay. And then to minimize the generation of hazardous waste management program are designed to meet or exceed compliance. Okay, kalau kamu tengok sini, is to meet or exceed compliance of with national or local regulation. Dia kena lebih daripada tu je. Kamu tak boleh nak turun bawah kata okey tak apalah kita longgar sikit lah, relax sikit, tak boleh, because you are going to create infection. Kalau benda yang kamu nak kerja ni tiba-tiba jadi macam spread macam COVID ni, so it would be bigger issue. Kalau if you go and read through macam mana SARS-CoV punya spread dekat Singapore, one of the reason benda tu dia punya biological safety cabinet tak di-clean lepas dia buat eksperimen. So that was the main cause benda tu start to spread. Okay, you all can go and Google about SARS-CoV infection dekat Singapore which started from the not clean biosafety punya BSc, Biosafety Cabinet. So benda-benda kecil macam tu boleh wujudkan satu spread. I will also show you. Okay, nanti. So the responsibility falls into the person who's generating the waste. Kenapa saya cakap balik? Semua orang kena faham. The person who's generating the waste, they have the responsibility untuk nak buang benda tu secara betul. Okay. Okay, nanti saya akan jawab soalan ini juga on the broken glass. Okay. Okay, let's go through why is something is important. Benda ni kalau kamu boleh Google FMD outbreak in UK. Okay, ini adalah satu case study yang saya suka tunjuk because this is something very interesting. So please harap saya dapat attention semua orang to go through this so that you would see benda yang sangat kecil ni how could be could create a very big spread. Okay, FMD outbreak in August 2007, 4 site detect, okay, detected by regular testing by government department. Dia tak ada isu yang besar, tapi bila dia pergi buat regular testing, macam dia pergi buat field surveillance study yang dia regularly buat dekat sana, dia dapat tahu ada FMD outbreak yang berlaku. Okay, and it covers 21 km radius and the government has to compensate the farmers. Itu satu lagi problem. Dia kena compensate, and 21 km radius tu besar, bukan kecil. Tapi macam mana benda ni berlaku? Kalau you tengok bila mereka buat investigation and dia mendapati bahawa 2 makmal yang bersebelahan dengan farm tu, cattle farming tu ialah animal health punya tu dengan Mariel Animal Health Laboratory. Dia ada 2 makmal yang bekerja dengan FMDV, dengan virus tu. Kalau mereka pun buat hipotesis benda tu airborne but very unlikely. So FMD bukan jenis airborne. So macam mana dia nak terbang daripada makmal? And dia kata waterborne likely but flooding of the lab and transmission is not possible sebab benda yang farm yang paling dekat, farm yang paling dekat daripada makmal tu adalah 4 km. So makmal dengan farm tu adalah jarak dia 4 km. Macam mana nak flood? Asalkan macam kat Shah Alam tu. Okay. So, and then they suspect human transmission, biosecurity, and then they tengok very possible. Apa yang dia suspect ialah orang yang bawa, dia nak khianat kepada farm tu, dia pergi spread ke dalam farm. So itu yang dalam investigation mereka duk suspect benda tu dan dia buat. Tetapi lepas dia habis investigation tu, apa yang didapati ialah traces of the virus was found in effluent pipe from Mariel Animal Health Laboratory to the government treatment plant. Maksudnya daripada makmal tu punya effluent pipe, sewerage system pipe tu kepada government punya, kepada macam contohnya Indah Water kan. Sebelum dia nak sampai ke Indah Water punya main pipe untuk ditreat, okay, dia ada traces of FMD. Okay, tetapi makmal tu sebenarnya dia kena treat dulu dia punya tu. So apa yang dia buat? So makmal tu dah ada in-house effluent treatment dengan citric acid disinfection. Citric acid disinfection is commonly used for FMD virus. So the FMD virus tu akan mati. So dia pun buatlah benda tu. Tak salah. Tapi FMD virus yang dia duk grow dalam makmal dia tak mati. Dia jadi resistant kepada citric acid. So itu yang benda yang penting yang dia tak realise. Makmal tu tak realise yang dia tak buat dia punya investigation, maksudnya dia punya due diligence. Virus yang dia duk kerja ni mati ke tak pakai, kalau kita pakai disinfectant tu. Mungkin strain virus yang dia pakai tu is resistant to citric acid. Dia tak mati. Dengan ni dia ada dalam effluent pipe. Tapi effluent pipe dia bukan atas jalan, dia bawah tanah. So kalau bawah tanah and effluent pipe, Apa masalahnya? Sebab pipe tu akan terus kepada main plant yang di mana dia akan treat dengan klorin lah apalah untuk nak semua akan mati. So what happened? Ini satu lagi case. There was a rupture of underground pipe caused by the root of a tree. So akar pokok tu, bila dia akar tu duk membesar, pipe tu pecah. Underground pipe tu, sebelum dia nak sampai kepada macam indah water punya tempat, pipe tu pecah. Dan pipe, apa yang jadi? Leakage of the water contaminated with the same FMD berlaku. So, benda tu pecah dan benda tu leak ke atas. Okay, maksudnya dia dah pancut keluar ke atas. And the rupture is nearby to a road to a nearby farm. So, maksudnya bila sebelum nak masuk ke farm tu, rupture tu ada ke depan tu. So orang-orang yang nak masuk semua. So apa yang mungkin berlaku? So how it was transmitted to other farm? Sebab lori dan benda yang dok bawa, lori lah, kereta lah, orang yang dok pergi ulang-alik ke farm dan lori yang hantar makanan from one farm to another farm. So dia pergi masuk dan dia transmit. So while it is moving, benda virus tu melekat kepada tayar dia lekat balik kepada farm tu punya, dan dia sampai ke farm tu dan it starts to transmit. So kalau you tengok benda yang very simple, okay, which is dia kena treat pakai citric acid tapi treatment tu tak jalan, and then you kata kalau underground pipe je tak ada masalah, tapi bila pipe tu pecah and air daripada pipe tu keluar kepada to the surface, and then dia start to transmit. Okay, and then kalau tengok ini adalah laporan dia, faulty pipe blamed for UK foot and mouth outbreak. Okay, so this was a very classic punya yang saya nak tunjuk, di mana a waste management, kalau waste management dia betul, kalau dia dah disinfect, pipe pecah ke tak pecah, no problem, benda tu tak ada masalah, tak ada apa-apa outbreak yang jadi. Okay, so you can go and read about that untuk nak lagi fahamkan. So kalau kamu tengok component of an infectious waste management, yang penting ialah kamu kena designate if the waste should be managed as infectious. Maksudnya macam tadilah, kita cerita pasal gelas kan. Semua orang tanya, glass slide macam mana? Glass slide macam mana? Glass slide macam mana? Glass slide infectious ke non-infectious? Sebenarnya dalam glass slide tu, sebelum kamu nak embed semua benda tu dah mati, bakteria ke apa semua dah mati. Kalau glass slide yang kamu gunakan live culture virus tu, kamu kena disinfect dulu. Okay, letak glass slide kamu kena rendam dalam disinfection before you want to throw. Okay, so sebenarnya glass slide you cannot letak dalam shark bin. Okay, so glass slide with bacteria smear you kena disinfect dulu, bacteria smear sebelum kamu nak buang. So they have procedures. We opt to choose a simple procedure. Gelas tu kita buang terus dalam shark, tak ada. So we have to, when you are doing with bacteria smearing, you kena disinfect dulu bagi bakteria tu mati, and then you dispose it as glass waste. Okay. Sekejap, ada soalan di sini ya. Oh, panjangnya soalan. Laboratory saya baca okay, kalau dia ada, I would like to ask about needle used to infectious live bacteria, should it be disinfect? Kalau dia ada needle, kamu tak boleh nak disinfect semua sebab you cannot handle, sebab dia akan create needle prick. Needle prick injury is another big thing. So sebab tu dia ada bin yang khas, sharp bin tu, macam you benda yang masuk dalam tak boleh nak keluar. So you just discard. Yang tu tak apa, sharp bin tak apa. Okay, itu satu. Laboratory need to adhere to specific design. Apa tu lari? of waste management before effluent was discharged to the environment, or standard waste treatment is enough to prevent incident occur? How about hospital laboratory where all sewerages go to central treatment plant? Okay, sebab tu kalau kamu ada makmal BSL 3, Biosafety Level 3 punya makmal, you akan ada special effluent system. Kalau bukan makmal BSL 3, you just handle makmal BSL 2, you tak ada effluent system. Sebab tu sebelum kamu nak pour your bacteria culture ke apa dalam sinki, kat Malaysia masih allowed to pour dalam sinki, but you need to treat it first. Maksudnya you ada bacteria culture tu, contoh dia macam ni ya, kalau kita tengok tangan saya botol air ni, assume this as bacteria culture, you tak boleh tuang terus dalam sinki, you kena treat dia dulu. So macam mana nak treat dia dependent dengan apa bakteria dia. Okay, ayat saya lagi sekali ya. Apa bakteria dia dengan berapa contact time. Later I will come to liquid waste. Saya akan explain juga. Contact time dia bukannya maksudnya sebab kalau saya guna sporulating bacteria, saya letak 10% sodium hypochlorite dan saya kena tunggu 30 minit untuk dia betul-betul mati before saya pour. Bukan 5 minit cukup dah. So you need to know bakteria apa dan treatment apa yang betul untuk dia. Okay. Okay, nanti yang mixed waste tu saya akan explain nanti. Okay, I will come back. So saya ada slide untuk mixed waste. So this is how, and when you go through this, so waste management process yang penting ialah kamu kena ingat step ni, 5 step ni, which means that you kena segregate, you kena pack, you kena label, kena ada handling, storage and untuk nak buat treatment. Okay, so first thing is segregation. Apa maksud segregation? Okay, macam kita cakap tadi lah. Okay, gelas kena masuk dalam gelas. Kalau benda tu infectious kepada infectious, non-infectious kepada non-infectious. Okay, so macam mana kamu nak buang benda tu is dependent. Kalau kamu baca go through the guideline, dia ada details. Okay, so kamu nampak tadi SOW 404, SW 401, dia ada benda-benda yang you kena buang. Okay, sebab dia perlukan treatment yang berlainan. Okay, liquid waste pun lain. So segregation is important. So sebab tu saya letak segregation is the key to any effective management. Without effective segregation system, the complete waste stream must be considered hazardous. Okay, katakan kamu ada bin hitam yang besar. Tiba-tiba kamu terbuang satu culture plate dalam tu. Sekarang, you kata tak apalah, aku ambil keluar je. Letak balik dalam bin kuning. Tak boleh dah. So the whole bag is now need to be treated as infectious waste. Sebab kamu tak tahu benda tu dah contaminate ke tak lagi. You cannot be doing that. So segregation is a very key sebab setiap benda yang kita letak dalam yellow bin tu, kamu kena faham, you need to pay for the treatment. And they count it by kilo ke apa ke, dia ada. So hospital senang sikit sebab dia dah ada kontrak. Kalau makmal, dia kira kilo. So treat it properly. Empty plastic chemical container should not be disposed in yellow bin. Betul. Dia chemical container. Kenapa kena dispose dalam yellow bin? Tak boleh. Yellow bin is for biological. Okay. So create waste stream to ensure you only treat what you have to. Ini penting. You see? Bukan semuanya. Each type of waste is treated appropriately. Only decontaminated what you must decontaminate. Okay. So tengok video ni kejap. Semua orang dengar ke suara dia? Can you all hear the sound from the video just now?
No sound, Doctor.
No sound Sorry. Saya akan pergi kepada next. So, tak apa, yang tu saya nak just terangkan jenis-jenis waste macam mana kita nak segregate. Sorry, I think video tu tak keluar sound because it's not connected. Tak apa, sekarang apa yang kita tengok ialah where to throw. So, benda yang kamu nampak ni, ini segregation. Segregation ni betul ke? Okay. Apa yang kamu nampak ni adalah bin apa? Yellow bin ke sharp bin? Can anyone? Tak boleh. Tak dengar. Okay. Okay. Okay, so benda ni tak boleh dibuat. Kenapa? Dia sharp bin, tapi gelas ni bukan sharp bin. Okay? And then this is sharp bin. You cannot be throwing your plasticware dalam sharp bin. So the segregation is bad. Okay, so you must know where that you need to throw your items. Okay, and then now packaging. So how you need to package it? Sekarang kamu tahu. Okay, so walaupun kamu ada yellow bin, tapi the yellow bin kamu tak boleh nak buang benda tu dalam. You need to have a yellow bag. Okay, kamu kena ada yellow bag, and then kamu kena letak autoclave bag. So yellow bag ni kamu letak dalam autoclave bag before you autoclave. Okay, so before you autoclave, kamu letak dalam yellow bag. Sorry, autoclave bag, so that you can autoclave that. Kenapa kamu tak boleh nak autoclave pakai yellow bag? Sebab yellow bag ni is non-autoclavable, tak tahan. So that's why you need to put it inside. You autoclave before you nak dispose. So the packaging tu is quite important. So ini adalah sharp waste. Ini adalah sharp bin. Okay, dengan untuk sharp sahaja. So kamu ada lid yang ni supaya benda yang masuk tu tak boleh keluar. You tak boleh nak ambil balik. Masuk tangan nak ambil balik. Okay, and it is closable, and when closed are not reopenable. Okay, kalau kamu tengok macam ni, benda sharp bin ni dah dibuka. So benda sharp bin yang dibuka ni tak boleh pakai dah. Okay, it is not safe sebab benda tu dah longgar. It must be quite tight. Kalau kamu force dia boleh buka lah. Tapi you shouldn't be forcing it to open. Okay, and it must be puncture proof. Maksudnya dia tak boleh nak berlubang ke apa bila ada needle dalam. Container size and location are critical. Okay, maksud dia size dia jangan pula bila you ada kerja kamu sikit, kamu letak container yang besar sampai 3-4 bulan baru kamu nak clear. Sebabnya benda tu infectious, dia akan start grow dalam ni, you tahu. So don't think that okay, lagi baik saya beli besar boleh accommodate banyak. Betul. Tapi when your work is not much, so beli container yang kecil so that you dispose it quite frequently. Benda tu kalau kamu biar macam tu, dia akan start to grow things inside, and benda tu akan start to fly. Okay, so and then avoid overfilling. Okay, ada orang tanya, if clinical sample is collect and put inside the autoclave bag, and after autoclave dispose dalam bag kuning boleh ke? Boleh juga. You can do like that. Tapi bila you nak letak autoclave bag ataupun kat satu tempat, if everyone knows that this is how you are going to do it, okay, kalau orang yang baru datang dia tengok bag tu warna biru, mungkin dia tak masukkan sampel dalam tu, dia nak cari bag kuning. So ini semua SOP saja, tak salah, but must make sure that everyone understands how you do it. Benda tu betul juga. Okay. Okay, sebenarnya benda-benda cartridge, COVID test kit semua-semua tu, you akan buang dalam yellow bin. Lepas you buang dalam yellow bin, kalau kamu ni makmal, you kena autoclave dulu sebelum kamu pergi letak kepada bin di luar bangunan yang akan diambil oleh service provider. Semacam makmal saya, the service provider comes in tiap minggu ataupun 2 minggu sekali. Because kalau tak cukup banyak pun dia tak datang sebab dia nak penuhkan lori dia. So benda yang saya nak letak kat luar tu, saya kena autoclave dulu sebelum saya pergi letak kat luar. Kalau hospital, you just go and dump it tanpa autoclave sebab benda tu di-collect every day. Okay, I hope that you all understand that. So ini adalah pasal needle prick, kenapa kamu tak boleh nak buat. So ni according to MOH. Daripada 1998 hingga 2005, 74.9% of injury was caused by needle stick injury. Sebab tu kamu tak boleh nak disinfect needle ke apa, kamu kena terus buang dalam tu. Kalau in the US, each year there are some 800,000 incident where people are accidentally struck by needle and sharp. The most common time of risk of spreading blood-borne pathogen occurs recapping needle. Sebab tu kita tak allow, hospital tak allow for you to recap. Failing to dispose used needle properly in puncture resistant sharp container. Sebab orang yang nak collect ke apa, dia akan kena injury tu. Kalau kamu sekarang puncture proof, dia bawa je bekas tu, dia pergi incinerate. So accidental breakage of tubes used for collection of blood in variety of healthcare setting. There are estimated 2,800 injuries from this glass capillary tube breakage every year. Sebab tu kalau you tengok sekarang, benda-benda yang dia collect untuk blood semua dah tukar jadi plastik dah. Dulu memang glass, tapi sekarang semuanya ditukar ke plastik sebab dia nak avoid benda ni juga. Okay. And it's easy for incineration. So kalau kamu tengok ni adalah liquid waste. So selalunya kamu prepare 10% bleach solution, kamu add 1:1 volume. Bukan 10% dalam ni. Solution dia 10%. Kalau ini 500 ml, kena add solution ni 500 ml. And then kamu kena tengok contact time. Saya kata at least 10 minutes. Generally, dia 10 minutes. Okay, tapi kalau kamu guna sporulate bakteria yang sporulating bakteria, dia kena tunggu lagi lama. Okay, so and then kamu boleh tuang dalam sinki. Okay, ini penting ya. Bukan ambil ni letak sikit je dalam ni, kacau dia, kata dah cukup. Tak. Okay, ini yang kebanyakannya orang buat salah. Okay, it has to be 1:1 volume. So labeling, lagi satu benda yang penting adalah label. So all benda-benda yang kamu kata ni kamu memang kena label. Okay, ini yang saya kata SW404 ni sisa klinikal dilupus melalui incinerator. Semua ni dah ada dalam guideline DOE yang 2009 tu. Okay, especially people who are working and dealing with this in hospitals, they are a bit aware than makmal. Tapi makmal pun kena ikut peraturan yang sama. Bukan kata makmal ada peraturan lain. Okay. Just tengok dulu ni. Ini adalah tempat yang dia nak dispose dan incineration dia macam ni. Ini tempat yang dia bakar. So dia buang macam tu je, dia bakar. Sebab tu saya kata gelas tidak boleh buang di dalam tu. Okay. So if you are still going to put the glass, you will spoil the whole big incinerator. Okay, lepas dia bakar tu benda-benda yang dah remaining dia akan scrape keluar. Tapi kalau gelas, benda tu akan start lekat pada dasar incinerator tu. Okay. So I will share these slides. So hopefully nanti you boleh tengok video tu juga. Okay, I can share the PowerPoint macam tu saja. I shared PDF, tapi sebab kamu orang tak boleh tengok ni, I will share the PowerPoint also. So, bila kamu kata nak storage, sekarang apa problem kat sini? Apa yang kamu tengok sini? Ini tempat yang saya pergi inspection. Okay, I cannot say kat mana, tapi ini tempat yang saya tengok inspection. Okay. So, apa yang kamu tengok kat sini? Bercampur, okay? Improper ventilation, chemical and clinical waste together. Benda yang penting, benda ni tak boleh. Okay, it has to be separated. Okay, clinical dengan chemical kena separate, tempat yang asing, it is required. Under CHRA, dia ada requirement sendiri. Okay, and under clinical waste, dia ada requirement sendiri. So, tak boleh nak campur. Kata, oh tempat waste kita sama, tak boleh. Okay, dia ada problem. So, benda ni leakage. Dan dia akan create more problem sebab dia akan ada mixed waste nanti. Okay. So, treatment and disposal. Satu yang penting ialah autoclave. Okay. And then incineration. Ada orang tanya ke dalam satu store waste tapi ada bin untuk clinical waste boleh ke? Tak boleh juga. Sebab dia kena separate. Sebab dia memang tak nak benda tu bercampur. Kalau ada campur, kalau ada leakage apa-apa berlaku, lagi susah nak handle. Okay. So, what happened kalau infectious waste kamu ni was thrown in domestic waste? So, apa yang akan berlaku kalau benda tu di-throw secara domestic waste? Infection kat orang ramai. Macam mana dia boleh jadi infection kat orang ramai? Water system contamins, betul. Okay. So, apa yang akan berlaku ialah kalau kamu ikut mana sampah kamu pergi, ke mana dia pergi, siapa yang akan handle sampah kamu tu, siapa yang ada akses kepada sampah ataupun yang benda yang ada contam tu pergi, ialah benda tu akan sampai kepada landfill. If it is wrongly done, benda tu akan sampai kepada landfill and dia jadi macam ni lah. Dekat landfill ni orang kerja. Beside orang, dia ada vektor-vektor, banyak vektor yang boleh transmit your bacteria ataupun infectious tu spread through. And then ni adalah spot-spot merah ni semua adalah landfill yang kamu boleh dapati di Peninsula Malaysia. Banyak landfill, bukan satu dua. Okay, so and it can end up very bad. Okay, so kalau kamu tengok landfill ni adalah predominant waste disposal in developing countries and it can cause groundwater contamination, air pollution, pest and health hazard kalau benda tu terbuang di dalam domestic waste. So handling of infectious waste tak pergi ke domestic waste and then tak apalah yang attitude tidak apa tu tak boleh ada because some people are trying to find living at the dump landfill. Mereka pergi collect sampah, mereka nak buat recycling and so on. So daripada infection daripada dia akan transmit kepada kawan-kawan dia, dekat family dia, and then that thing will just spread like how COVID spread. So it can be a very big issue. Okay? So key messages ni adalah waste should be segregated in an appropriate way. Different method for collection and storage mesti kena ada for biological waste are necessary different type and there are different treatment method that are appropriate according to the risk and the type. Okay. So thank you. Tapi sebelum saya nak habiskan, saya ada few soalan yang saya nak address kat sini. Ialah orang duk tanya clinical and chemical waste kalau dia mix macam mana. Okay, so kalau clinical dengan chemical waste mix, apa yang you kena buat? Mana yang dangerous dulu? So the risk analysis has to be done. Okay. So risk analysis has to be done. Maksudnya yang mana yang lebih teruk. Kalau chemical tu lebih teruk, macam dia fume, dia berikan spread, treat chemical dulu, then treat your infectious. Tapi selalunya infectious dulu. So bila dia campur, and that chemical solution tidak boleh disinfect your bacteria ke apa yang tercampur tu, disinfect dulu dengan chemical, then treat it as liquid chemical waste ke apa. Okay, kalau chemical tu macam contohnya kamu ada chemical waste formalin, bila kamu masukkan bakteria dalam tu, bakteria tu dah mati dah, tak boleh hidup dalam formalin, and then you directly treat it as chemical waste. So you kena buat risk analysis, yang mana yang risiko dia tinggi dulu. Macam contohnya ada juga isu yang saya pergi satu makmal, dia ada buat radioactive labeling. Dia kata saya nak treat yang mana satu? Saya kata radioactive dulu. You treat it as radioactive waste dulu. Sebab automatically benda tu akan mati, betul? Tak juga. Sebab dia treat as radioactive sebab dia pakai P32, 2 minggu je. So half life dia 2 minggu, tapi berapa lama dia kena tunggu. So dia kena masukkan disinfectant dalam solution tu and then treat it as radioactive waste. Okay, sebab P32 tu tak membunuhkan bakteria, tapi benda itu semua dia akan buat dalam dia punya safe environment untuk P32 tu. So dia ada juga radioactive waste yang melibatkan dengan sampel. Okay, I think I answered. Saya rasa saya ada lagi satu. Hold on. Ada orang tanya, must all human sample be autoclaved before disposal? Human sample macam blood, bodily fluid, semua-semua tu, kalau makmal perlu sebelum kamu nak dispose. Kalau hospital, as I said, it will be collected every day. Kalau pathological waste, dia kena ada dalam, you kena tengok macam mana you nak dispose pathological waste tu. Okay? So dia ada prosedur untuk nak dispose pathological waste. So you can refer to pathologist. So they can give you, sebab kalau dia non-infectious, dia ada cara dia lain. Okay. Lab kecil tak ada autoclave. Kalau nak pakai water bath boleh tak? Boleh. Boleh. But it has to be, timing dia tu kena penting. How you do it tu kena penting. Kalau tak, you kena pandai-pandai collect dia and then you hantar ke tempat sebelah ke mana yang ada autoclave ataupun terus ke incineration. Okay, must all sample be autoclaved before disposal? Other labs do not, only dispose in yellow bin. Tak, semua sampel itu kata hospital setting dengan makmal yang bukan di dalam hospital lain. Hospital setting every day you akan dispose dan every day ada collection. So tak autoclave, tak apa, I'm repeating this. Kalau makmal yang you don't have everyday collection, all of it must be autoclave before kamu simpan sehingga service provider datang untuk ambil. Okay. Okay, saya rasa saya dah jawab soalan-soalan yang lama tu. Hold on, saya check balik ada soalan tak. Panjang juga. Hold on.
I think there are a few more questions. One person asked how to dispose the test COVID that is done at home. The second one is there any recommended disposal procedure like double bag and so on? And the third question is, if a hospital engages vendor for clinical waste disposal, do the microbiology lab waste need to be treated before disposal? I think these are the latest questions.
Regarding the home test kit, okay, there's a very big issue when the home test kit came out, and we also have written to MOH telling that this has to be sent when the sample is positive, when your test is positive, sebab dia boleh spread. Okay, usually when your test is positive, benda tu kena dimasukkan dalam boiling water, then for about 15-20 minutes baru dia keluarkan dan dibuang. But now they are saying that just tape it and buang. So the procedure is a bit bad. Tapi apa yang mereka kata, there's no other choice that apa yang mereka boleh buat. So during that time there was a very big debate on that. Tapi benda yang patut dibuatnya, you might have seen people encouraging people to use boiling water, rendam dalam air didih sebelum dia buang benda tu. That has to be done at home, the best thing. And then if hospital engage a vendor for clinical waste disposal, do the microbiological lab waste need to be treated before disposal? I'm repeating it back. Kalau your waste is being collected on daily basis and removed from the hospital on daily basis, tak perlu. But you can't manage to autoclave everything before. So sebab dia everyday benda tu akan pergi. Kita nak autoclave benda tu dengan alasannya macam saya kata makmal saya, the service provider comes in once a week ataupun 2 minggu sekali. So kalau saya tak autoclave, saya ikat beg kuning tu, saya letak kat luar, bakteria tu dah start grow dalam beg kuning tu. And then bila orang nak handle tu, it is becoming more hazardous. Bukan satu jenis bakteria, banyak dalam tu. Tapi if it is every day you dispose, every day is collected to be sent for incineration, tak perlu. Just make sure that the yellow bin is completely sealed and ketat. Macam ada orang tanya, double bagging tu perlu ke? Perlu. Kalau you boleh buat first bag, you dah ikat ketat-ketat, then second bag, you ikat ketat-ketat, then you dispose, lagi bagus. It's all SOPs and procedures saja. I'm repeating again, everyday collection, tak payah, tak perlu. That's why hospital don't practice autoclaving before disposal. Okay. Ya, ada lagi. Okay,
Doctor,
what
there's
is
another question. Yeah, another question on the glass disposal. Maybe you can explain again, Doctor.
Yeah. So I Google for broken glass disposal box, very expensive. Broken glass disposal box semua tu tak payah. Okay, there's no need untuk beli. Okay, you ada satu container ke styrofoam punya box ke apa yang kamu ada tu ada je. And then tapi you kena, kalau benda tu dalam, contohnya botol ni adalah dalam culture bakteria jatuh pecah. So what you need to do, you selalunya you treat it as spill, kamu tuang dulu disinfectant, then you collect all the pieces of glass, you put it in a container with disinfectant, lepas tu you drain out dan collect semua glass tu, letak dalam satu container box yang kamu akan dispose. Yang ni Mak Saleh punya, dia punya requirement tu tinggi, dia kena ada ketebalan lah apalah, kita tak ada. Just to make sure that it is out of reach dan container yang kamu simpan broken glass tu is intact. Itu je. Tak perlu pergi beli mahal-mahal punya semua tu. Okay. Okay, how to dispose broken tempered or amber glass since recycling company usually do not accept it. Okay. I will check on this sebab benda yang kami ada tu, company tu dia amber glass pun dia ambil. Sebab dia ada, dia depends on recycling company. Tempered glass dengan amber glass ni cara nak recycle dia berbeza. Sebab tu kita kena separatekan normal glass, tempered dengan amber glass. Kita punya segregation tu kena betul. So then they will accept it. Kalau kita campurkan semua, selalunya dia tak accept sebab cara recycling and processing is a bit different. Okay, I will try to get these details and also will try to share with you all. Okay, if animal tissue culture waste, should we need to autoclave even though already disinfected? Tak payah. Kalau tissue culture waste tu, you dah disinfect dengan Clorox, benda tu akan convert jadi liquid waste. So kamu buang. Yang plastic waste yang di disposal tu, kamu rinse dengan disinfectant tu dan letak dalam yellow bin. Dan kalau daily, you buang. Kalau bukan daily, you still need to autoclave. Okay. Is there any more soalan?
Okay, if the participants still have any question, please kindly unmute your mic or you can post it in the chat. I think they were very happy, Doctor, for your topic today because it's close to our hearts for the laboratory personnel. And I think for the those who are working in other laboratories, not only the hospital laboratories, but I think a few other categories. I think food laboratories, animal, and also I think this is quite an interesting topic and it's a very wide topic, I would say. Even 2 hours won't enough, but we try to concise it in 1 hour plus webinar today. So I hope our participants has benefited from our topic today. And I suppose there is no more questions since they were asking while you were presenting the webinar. So we will try to share our slides and also the webinar recording today. And just to inform everyone that today's webinar, we had 422 participants, Doctor. So that's quite a number. Quite a number, and it's quite an achievement for us in the PPSM as well. So thank you for all the participants who has actively participated, asking questions to you. And I hope our participants would fill up the feedback form and also the attendance form for you guys to get the e-certs. And we are looking forward for your feedbacks on the next hot topic from you guys as well. And thank you, Doctor, for your time and knowledge. And we hope to see more from your association as well. Thank you so much. Thank you, everyone.
Thank you so much. And then I think, Mrs. Vidya, I think you can share the slides with the participants, no problem, because they are asking whether the slides can be shared.
Yeah, we will share it later in our website, and also the recording will be available later.
Okay, thank you so much. Hope that everyone get informations, the message that we want to deliver. Thank you.